Okay, so I'm a PhD student at Uppsala University in Sweden, and today I'm going to be talking about a little side project that I did describing tiny acritarchs, which are sort of remarkably small even on the scale of microfossils. So first of all, I'm going to give you a brief overview as to what organic walled microfossils are and what they're doing in the Phanerozoic and why do we care. Then I'm going to be talking about geological background to the Lükati Formation in Estonia, where the material comes from. I'm going to show you some of the new species, new genera of these particularly small acritarchs. I'm going to be talking about their biological affinities and eventually what this means for the evolution of phytoplankton at the beginning of Phanerozoic. Right. So for most of the Earth's history, there was sort of no biomineralizing organisms. So pretty much everything that we see in the Precambrian in the Precambrian is sort of organic walled. However, what we mean by organic walled microfossils in sort of this case is sort of these strange unicellular forms which have a morphology slightly more complex than your usual bacteria or just bacterial sheets. So we're assuming there would be some kind of eukaryotes. And a lot of these organic walled microfossils, for a long time, they've been called acritarchs, which means of uncertain origins. And if we're going to be progressive about this, it's a pretty sort of useless term because a lot of these morphotypes were taken out of the unknown useless acritarchs and placed into groups, identified and placed into known groups. For example, some stem eukaryotes, some kind of protists, green algae, plant spores, fungi from Precambrian, and so on. So what are they? We know they're sort of unicellular. They have organic wall, which may have various morphological elements, and they're eukaryotic, meaning that they would be a bit more complicated than you would expect from a bacterial fossil. They have a very long stratigraphic range from Proterozoic to recent, and they occur in aquatic environments. So how would these organisms be formed essentially? And sort of recent work suggests that most of these sort of forms that we see here are a result of the cyst formation as a protist will sort of go into the resting or reproductive stage. And just to make things more easier, in the Cambrian and for most of Precambrian, we are dealing with sort of two main morphologies. Either they have processes, they're called pentamorphs, or they lack processes and they're just simple spheromorphs. And they have certain diagnostic characters which help us to define specific groups. So those characters would be processes, some kind of wall structure, whether they have the excisment opening or not. And it's quite important to know that they're all acetolysis resistant, meaning they're quite recalcitrant and resilient. So this probably points to some kind of primary feature of the vesicle. So for most of the acritarchs history, they were present in Mesoproterozoic, Neoproterozoic, but not in great abundance. We see an explosion of different organic walled microfossils at the Cambrian, which is sort of a counterpart to the Cambrian explosion. So it wasn't just the evolutionary explosion of metazoans, but also of these tiny little forms. When it comes to the cell size of these things, throughout most of Precambrian, they were quite large, and we're talking something like 200 to maybe 500 micron in diameter. But then, at the beginning of Phanerozoic, there is this striking trend towards the reduction of the vesicle or of the cell size in organic walled microfossils, even to 10 micron across. Right, so my material derives from Lükati Formation in northern Estonia, exposed just west of Tallinn. This is the Kopli quarry, which is now unfortunately flooded for a couple of years now. You cannot actually access it. So most of my material was collected a couple of years ago. And this was a part of the Baltica paleocontinent, and it was a part of the Baltic syncline. It's a silistoclastic formation, which is very thinly bedded, and it's sort of alteration of very thin silt and clay layers, which are still very squishy. They're not very well consolidated. We found some evidence of that point to the shallow marine setting of the formation, such as the desiccation cracks and so on. And the formation was thermally and diagenetically unaltered, which is great if we want to study these microfossils, because they have been altered, so they still preserved very, very fine structures. And the formation is also rich in other polynomorphs and small shelly fossils. This would be the age of the Lükati Formation, from sort of 530 to 514 million years. Which was constrained by the trilobite zones and these sort of polymorph zones. Next. So the material was, I used standard palaeontological technique to extract the individual microfossils from the rock by chugging them into the hydrofluoric acid, leaving them for a couple of days for the rock to be dissolved, and then filtering them through and eventually mounting them either on glass preparates or SEM stubs. And this is some of the diversity from the Lükati Formation. We have quite a lot of spheromorphic acritarchs, even Prasinophytes, and also these acanthamorphs with quite long processes. We also found, and I was about to say that biological affinities of most of these fossils are sort of now well understood, and we're mainly dealing with Prasinophytes and chlorophyte green algae. And this is one of the new species that was discovered in the formation, while sort of, it was kind of a side project because I was looking to look at the formation to compare it with some of my other material. When this little fossil cropped up, and it's pretty blobby, very fluffy. You can't tell much just by looking at it with a light microscope. So underneath the SEM, it shows a bit more information. We see that it has this very corrugated, wrinkled wall, but it's very different from just compression wrinkles that we normally see in just kind of poorly preserved leospheroids. So this seems to be the original corrugation, and it's also very finely perforate. So if we zoom in on this microfossil, it has this very, very tiny pores, sort of maybe 25 to 50 nanometers across. Sometimes they can be stretched, which indicates the flexibility of the organic wall. And there are a lot of these sort of corrugations to the wall. This is also when I was describing the species and digging through the literature to see if there was anything similar. I found that there were some old papers actually illustrating the species by just describing it as a synovial cluster or unnamed cluster A or B. So even though this wasn't named previously, it's quite common mostly around the Avalonian Seaway on various paleocontinents, Avalonia, which is present today, which is Britain today, Iberia, Spain, and Baltica, sort of Norway and Estonia here. So have we seen anything similar in fossil record? Well, there was this very common Cambrian acanthamorphic acritarch called Asteridium, which is very, very similar to Reticella, the new species. And it has tiny, tiny thorny processes. So just under light microscope, it can be easily mistaken. So we need SEM to be able to tell these two species apart. Next, there are some counterparts with the similar size of maybe 5 micron across from the Cretaceous. And these were actually described as the earliest members of the picoplankton ecological grouping. Further comparisons, Reticella is very similar to corrugated acritarchs, some kind of wrinkled acritarchs, which are common in the fossil record from Neoproterozoic to Neogene. But usually these sort of wrinkled forms are much, much larger. For example, Cerebrosphaera is a couple of hundred micron across. It was only the Prasinophyte Pterospermella, which has this sort of internal body of the phycoma. It's the similar size and similar morphologies to Reticella. Then we have another Prasinophytes, which has these sort of porous walls. Again, it has very, very long range from Precambrian to recent. However, it's much larger than Reticella, and here we see one of the specimens of the new species and a full Tasmanites, also from the Lükati Formation. So far, Tasmanites and Pterospermella seem to be two closest, seem to have sort of most closely resembling elements of morphology to the new species, but they seem to be much, much larger. However, these two genera and fossil families are thought to be undisputed Prasinophyte algae, and they were identified so by Fenson in the 90s. So we have these elements of morphology that are very Prasinophyte. So I would conclude that Reticella corruagte would be a Prasinophyte alga. Right. So we have a particularly tiny Prasinophytes in the Cambrian. Why should we care? And mainly because this might be the first record of the picoplankton that we're seeing. So far, there was this very unimaginative term of small acritarchs coined for the tiny picoplankton members from the Cretaceous, which was supposed to be the first Prasinophytes picoplankton. However, if we look back to the Cambrian, there is a tiny diversity, it's not very well diverse, but there is a record which this new form enriches. And we have several occurrences of these small acritarchs from the Cambrian, Paleozoic, Mesozoic, and Cenozoic, telling us that this particularly tiny picoplankton was common throughout Phanerozoic. And we are currently in Uppsala making a little database of the small acritarchs to try and get some more quantitative data to talk about their evolution. Right. So as I've shown before, Reticella, here pictured in green, is quite widely spread. We have acritarchs of organic wall microfossils of similar size that are also present throughout most of the paleocontinents in the Cambrian, giving them, sorry, it's fine. Saying that we have a significant record of tiny little phytoplankton members. So we could have the first members of picoplankton already in the Cambrian. And why hasn't picoplankton evolved before if we are seeing Prasinophytes such as Tasmanites and Pterospermella in the Precambrian? So there was a recent paper last year in Nature Geoscience by Lenton, which said that maybe due to the evolution of sponges, they were filtering quite a lot of this tiny picoplankton, which maybe opened up a niche for more picoplankton than then Prasinophytes in the Cambrian were able to exploit. So for the summary, we have a diverse polynomorph record in the Cambrian Lükati Formation. Amongst them, a new species, which is, well, it's most interesting. The most interesting thing about it that it's so tiny. It shares characters with Prasinophyte algae. You need SEM to be able to identify it. And we have the diversification of tiny phytoplankton in the Cambrian, which could be the onset of eukaryotic picoplankton. And this new size class of small acritarchs was likely underestimated because you need SEM to be able to identify them. So we are currently compiling all the literature references to acritarchs, maybe 5 to 10 micron in diameter, and also making sort of a diversity plot of the Cambrian Prasinophytes to see how they evolved. So I would just like to thank my supervisors, Malgosia and Sebastian, as well as Heikki Bauert, who was an excellent guide when I visited Estonia, and all the people who gave me money. Thank you.